Okay, my group just focus on the bacterial disease, include the E. coli, Salmonella, Mycoplasma. Also, this is for the avian disease. Also, some bacterial disease in the cattle, such as the brucella. And now, today I will show some bacterial disease in the avian. So for the contents, I just focus on the two different diseases. One is avian disease caused by APAC, E. coli. Then second one is the Mycoplasma. The first one is Avian Colibacillosis. This disease is caused by the APAC, means avian pathogenic E. coli. And for this disease, it present a number of different things and different forms, including cause air sac disease. Also, sometimes is swollen head. Also, that many different things also can cause the airborne death. Also, previously we think that's a secondary pathogen followed by the virus infection. But now we know the APAC can be the primary pathogen to cause the disease, because when we compare the APAC, the genome to the commensal E. coli, the genomic is very different, because the pathogenic there has more virulent vectors and genes compared to the commensal E. coli. So this pathogenic can cause the disease directly, not be the secondary pathogen. So for the biology of the E. coli, I think most people will know the biology, because E. coli is located in our gut and everywhere. And because I show this, because just for the one-way isolation and identification for the E. coli, we should use some different medium, such as selective medium, Mycoplasmosis agar, also EMV agar. So the E. coli show different clones in this selective medium. So when we isolate, we just see the different clone and different color. We can know which clone is E. coli, which clone is Salmonella, and the other bacteria. Also, for the E. coli, there are different classification, but the cell type is the most popular classification. Is according to the O antigen, K antigen, H antigen, and F antigen. But the most popular is we just use the O antigen as the classification. And now just about 180 different O antigen identified. And for about 80 K and 60 F antigen. Then for the pathogenic E. coli, as I told before, although most E. coli, they are located in our gut and they are commensal, they are harmless to our health, but certain cell type or certain pathogenic E. coli can cause different disease from the intestinal or extraintestinal infection. For the APAC, it belongs to the EXPEC, and also when we compare the genomic sequence, the commensal E. coli, the INPEC and the EXPEC, they belong to two different paragraphs. Also now we know, because APAC is very close to the human EXPEC, such as UPAC and NMAC, and these two different EXPEC can cause the disease in humans, such as the NMAC can cause the baby to death, because the NMAC can enter into the brain of the baby, and then because the BBB, the blood brain is not very good, so the E. coli can cross the BBB and then cause some disease in the brain and cause death. And now we know APEC and human ESGC share many different for the genomic is very close, and also they share many same valence factors such as the and also share many same serotypes such as O1, O2, O78, and O19. So another, in addition, APEC, now we always use antibiotics to treat the APEC infection. So the APEC can carry many antibiotics resistant genes. Then when we eat the food contaminated with the APEC, then the antibiotic resistant gene will transfer from the food to human, then will cause the human infection. Also, for the world, the distribution of the APEC is very worldwide distributed, and also different. I also collect different articles, and now most APEC belong to O1, O2, O78 is the popular serotypes of the APEC in the world, but different countries that has different permitted popular O serotype, such as in China. In these days, O148, 145 may be the popular serotype now, but before it's O78. Also, for another classification is MRST. The most common ST profile of APEC is ST117, ST140, and ST95. But this type also can cause some disease in human. Also, another classification is phyloglops, but for the APEC, there is not it's not most popular. It's for each group is similar, the presentation. Also, another one is the presence of the antibiotic resistant genes is well present in the APEC. And in China, we just isolate more than 5,000 different APEC strains, and then we identify the cell type, and also detect the virus genes, and also we test the drug resistance. And we found most of the APEC belong to O78. This is about half of the serotype, because as we know, the APEC there were about 180 different serotypes, but O78 is the primary, is a popular serotype, and also the APEC belong to B2 and A. This is some more than group D and E. Also, for the APEC, why we detect the various genes? Because for the commensal E. coli, the harmless, and the pathogenic E. coli, compared the pathogenic E. coli, they cover many different variants genes. Then the E. coli, pathogenic E. coli use this variance genes to cause the disease. So we detect the variance genes. So most of the APEC have the many different variance genes. Also in China, the APEC show multi-drug resistance. We test about 40 different antibiotics, but some strains can resist to almost all the antibiotics. So if we use these antibiotics to treat, so it cannot treat because it resistant to the antibiotics. And for the pathogenic mechanism of the APEC, because we should know how the APEC causes the disease, so we just focus on the step of the pathogenic how to cause the disease. Then first one, the APEC enter into the body of the avian from the respiratory and also sometimes from the oral. Then after the colonization, invasion, and then enter to the bloodstream and go to every tissue of the body. Then also we found that the APEC can enter into the brain, can cause some disease in the brain. And this is similar to the human EHPEC, AMEC. And during the infection, the various factors play important roles in the infection, in the process of the infection of the APEC. So include the security system and the OMV and RPS and other various factors. For our lab, we identified many different various factors, but I just focus on the secret system. In the gram-negative bacteria, there was 6 different assays. So from the type 1, type 3, type 4, and type 6 secret system, they can just secrete the protein directly into outside of the bacteria or just into the host cells. But for the type 5 and type 2 secret system, there was 2 steps. They need the protein should be through to 2 types. And this just some results of our lab. For the type 3 system in APEC, for our lab, we are first found that type 3 T-cell, because in E. coli there were 2 different type 3 secret systems. But for the first one is located in the EHEC and EPEC. But the second one is ETT2. We call the E. coli type 3 secret system 2. Then we found this system is wide distributed in APEC. Also, we find 5 different S form of the system. And this before our publication, there were no information about the ET2 in the APEC. So also we found the ET2 locus is present in the popular APEC serotype 078, 01, and 02. Also there were, because before they think ET2 have some gene deletion and gene mutation, they think ET2 is non-function. But we found there was also intact ET2 locus also located in the APEC and also located in the AMEC. This ET2, so it indicates ET2 can be functioned in the APEC. Also, we do the gene mutation and also accompanying use the red system and found the core factor of the ET2, EIVC, is ATP and contribute to the bacterial motility, serum resistance, and survival, and the valence of the APEC. Also, another regulator in the ET2, ETI, also can affect the gene expression and contribute to the bacterial valence. And also another regulator, not located in the ET2 locus, in the other located in the other genomic. They also contribute to the valence of APEC through regulation of the ETT2 expression. But this is some of the results of the function of the type 3 system. Also, we want to determine the function of the type 5 system. It also we identified 5 novel T566 in APEC. This 5 T5 60 SS is we found first. We use different methods. For example, we sequence the genome and of the bacteria, then compare the commensal E. coli and the pathogenic E. coli. Then we found the new and the novel type 560 SS, and then we also generate the mutant strain and the pathogenic strain, and then to compare the motility, adaptation ability, and other variants, and found these 5 different T560 SS play an important role in the valence of the APEC. Also contribute to the colonization in the duck, and also in the chicken, and also in the mouse. Because we use a different animal model. And another system is type 6 SS. We found first in this type 6 SS, which I identified in 205. But in that time, we don't not focus on this system. After that, then we because type 6 SS is found in 206, but in 205 we found already, but we at that time we didn't focus on that. And we also our results show that type 6 system is played in key roles in the APEC variance, and also some regulator CPXR. This also can module the expression and facilitate the interbacterial competitive action and the valence of the APEC. And because for the system, for the security system, they can develop some effector protein into the outside the bacteria, also inside of the host cells. So we also use the different techniques to identify novel effectors. But for that study, we are continue to focus on what's the function of the effector, how this effector subvert the host immunity. But didn't finish. So maybe next time we have the full result, I will show you. Also, APEC can enter into the brain of the body. So we also focus on the emission of the APEC. So we also identify some emissions such as IBEA, IBEB, and IBER, and focus on how these proteins functions in the APEC to enter into the brain. And we found that these proteins play important role in the invasion and the valence of the APEC. And this is just the transmission routes of the APEC. They can transfer from different routes, such as from the egg. Also, because now we also determine which how the APEC to cause the disease of the embryo. But we also focus on the salmonella. We found at the first 10 days of when the hatch of the embryo, so the most mortality of the embryo will be caused by salmonella. But in the last 10 days of the embryo death, may be caused by APEC. And then also this is just a clinical sense of the APEC infection, but as we know, APEC can infect different tissues such as head, heart, liver, and also feet, and also so we show different things. Because due to the time limitation, I will not just show you. Also, this is just some future of the APEC infection. Just this picture is when we do the infection experiment, we just take picture of our level. Also, is some future fingers. And for the diagnosis of the APEC infection, so the traditional is we just isolate the E. coli from the test avian. But how to know this E. coli is a commensal E. coli or pathogenic E. coli? So we need after the bacterial infection, and we need to detect the variance genes, because if the especially for the vessels, there was many commensal E. coli. So we think it's APEC, but this E. coli cannot cause the disease. So after we get the E. coli clones, we need to do the variance genes detection. For that, so we develop some multiplasmid PCR to detect the various genes, and we show that if this E. coli harbor 5 different various genes, then this E. coli can be think the is APEC. Otherwise, if the E. coli just have one various gene, so this E. coli cannot cause the disease, and this should be the commensal E. coli. Also, after detect the various genes, then we also use different strains to infect with the animals. Then the result same. If the APEC have more various genes, then they will cause more death for the animals. If there was just have one various gene, they harmless. Also, for the old serotyping, because as we know that there was 180 different old serotypes. So that but now we just use the specific O antigens antiserum, but this is costly and time consuming. So we develop different PCR method for the O serotyping. Also for the H serotyping, we just according to the special target of the genes, so different O serotype strains. For the management, for the control of the APEC infection, just first manage, we just decrease the APEC in the environment, in the water, in the feed, and also because and another one is antibiotic treatment. But now I don't know what's the situation in Pakistan, but in China, most of the APEC is resistant to the antibiotics, so it's very difficult to use antibiotics to prevent the APEC infection. Also, vaccines, there are different vaccines for the APEC infection. For killed vaccines, live vaccines, and also subunit vaccines. I just show you because time limitation. And also for the phage treatment, I also see the schedule. There also someone will give precision for the phage treatment. I also focus on the phage treatment for the of different bacterial infection, and we also isolate many different phages of different bacteria, and also to test the treat effect of the different bacteria, and they show good results. For the part 2 is Mycoplasmosis, and this is just some information of the Mycoplasmosis, and the most popular is MG and MS in the country, in China, also in the world. And because Mycoplasma is lactose cell wall, so when we select the antibiotics, so we should not select the antibiotics that affect the cell wall thickness, such as penicillin, because it's useless for the Mycoplasma. Also, this is the distribution of the MG and MS are they are all the world distribution. Also, this is some different subpopulation, and for the MG and MS, they are different. For example, MS was most prevalent in predators, but for the MG, it's in the hens. And in China, also, MG and MS most popular in the south and eastern of the China, not in the north of the China. And also there shows some different type. In our lab, we also isolate some many different MS strains, and we found the D-type MS caused disease. This all results. And for the pathogenic mechanism of the Mycoplasma, but now it's not very clear. But the important step is how the Mycoplasma is attached or adhesion to the host cells. This is very important. So for that, we also identify several proteins be adhesion. For example, this just show you these three different proteins play important roles to adhere to the host cells. And this transmission of the Mycoplasma, they can transmission from the hen to the egg, also can vertical transfer. Also, this clinical sense of the MG infection, just how to say, just show you. And for the diagnosis of the Mycoplasma infection, there are three different methods. For the first one, isolation and identification, but this one is time cost. Also, most level, even in China, most level they cannot isolate, because for the Mycoplasma is very difficult to isolation. Because the Mycoplasma, you need many high quality for the media. This isolation, but also in the chronic state of the infection, we cannot isolate. For the serum diagnosis, there was three different methods. For the SPA, ELISA, and HI. Also, these three different methods also have their advantage or disadvantage. But for lab, we also development of ELISA method, and this we also use special protein. We identify use different method, and also this we developed this kit already. Also, for the molecular biology detection, we just use the PCR, RT-PCR, and other method. We also developed different PCR method. Also now, for the MS, there was a vaccine for MSH strain from Australia. So, but now we don't know which strain. When we use this vaccine, we don't know whether this is the vaccine strain or wild type strain. So we compare the genomic of the MSH strain and the wild type strain. We found the SNP different. So we, according to the SNP size, so develop the SNP based problem method, and we can know which one is the wild type strain, which one is the vaccine strain. Also, we published already. And for the control of the Mycoplasma infection, is also we management also decrease the MS in the environment. For the antibiotic treatment, so we need to select the suitable antibiotics to treat. Also for the vaccines, we know there are also for the MS there was killed vaccines and lived vaccines, but for the MS and MG, the killed vaccines, the protect effect is not very good. But for the live vaccines, so we need to distinguish what's the vaccine strain or which one is the wild type strain. To this just show some antibiotic treatment. So because we need to select the different antibiotics. And also in China, now some farm use the traditional Chinese medicine to prevent the Mycoplasma infection. And this just vaccines, and this is vaccine from US, from Australia, and for the MS and MG, just show the list. And for lab, we just development of Mycoplasma submitted vaccines. Also, we find good results. Okay, for the final, it's just a summary. Is for the control of the impact of the Mycoplasma infection, and these two diseases are the popular respiratory vector disease of the poultry, and also they are well distributed in the world. For the control of the impact and the Mycoplasma infection, we need to show good management to decrease the impact and the Mycoplasma in the environment, in the water, in the feed. And also, most important for the bacterial infection, most we use antibiotic treatment, but we need select sensitive antibiotics for use. Another one is we use vaccine, but also we need to select suitable vaccines. Okay, thank you very much.